Hey guys, welcome to Dr. Varsha Gaitonde's channel. Here we shall discuss about some of the important questions related to seed science and technology. These questions are very important for competitive exams, especially for JRF and SRF. So let's begin. Here I am covering some multiple types of questions, true or false type, or some of the direct questions which may be in the form of match the following or something else. So, first question is, which one of the following is an essential factor for germination of a seed? Four options are given here: light, minerals, genetic makeup, and air, that is oxygen. For your convenience, I am reading all the options so that you just listen to the question and you tick or you write down the answer. After that, you can see what is the exact answer. And to first question, the answer is air or oxygen. The second question is, field germination is always lower than the germinations of seeds tested in laboratory. The question is whether this question or this statement is true or false. This is a true statement, that is, field germination is always lower than the germination of seeds tested in laboratory, because many of the environmental factors, soil factors will be influencing on the expression of the seed characters. The third question is, foliar feeding of nitrogen at the grain filling stage improves. Options given here are germination, initial growth, growth vigor of seedlings, and weakens dormancy. The best and suitable answer to this question, that is, foliar feeding of nitrogen at the grain filling stage improves, it weakens the dormancy. If you give nitrogen in the initial stage, means it gives better vegetative uh, characterization. Here here all the answers are true. That means foliar feeding of nitrogen at the green filling stage improves germination, initial growth, growth vigor of the seedlings. The fourth question is, weight of 100 grains or seeds in case of bold seeds like maize, and weight of 1000 seeds in case of small seeds like rice refers respectively to 100 seeds weight is known as seed index and 1000 seeds weight is known as test weight. We shall move to the next question. The fifth one is classes of seeds and their color of tags. Here, first one is nucleus seed. Nucleus seed has buff color. After matching, I am telling you, nucleus seed is having buff color tag. Breeder seed, as you know, this is very popular, that has golden yellow or golden brown tag. Foundation seed has white tag. Registered seed has purple color tag. Certified seed has azure blue tag, and truthfully labeled seed has opal green. For your kind information, I'll repeat the answers. Nucleus seed has buff color tag, breeder seed golden yellow or golden brown, foundation seed has white tag, registered seed purple color, certified seed azure blue, and truthfully labeled seed, this is having opal green color tag. The next question is matching the minimum isolation distance. Here the question is direct and answers are already matched. Try to remember this. The crops like cereals and pulses, rice, wheat, barley, oats, soybean and groundnut, which are purely self-pollinated crops, has isolation distance of 3 meter for foundation seed as well as certified seed production. Onion bulb or onion is having 5 meter isolation distance for foundation as well as certified seed. Tomato, it is having 50 meter isolation distance for foundation seed and 25 meters for certified seed. When we talk about okra or bhindi, this has 400 meters for foundation and 200 meters for certified seed. When we talk about castor, it is 300 meter for foundation and 150 meter for certified seed. Pearl millet or bajra hybrids are having 1000 meter isolation distance for foundation seed and 200 meter for certified seed. Cauliflower, cabbage, radish and turnip, which are popular cole crops, these are having 1600 meter isolation distance for foundation seed and 1000 meter for certified seed. Coming to rapeseed and mustard, here the criteria is entirely different. As you can see, for self-compatible and self-incompatible foundation seed, the distance is, or the isolation distance we may say, is for self-compatible is 50 meters and self-incompatible is 100 meters. Basically for hybrids or general rapeseed and mustard varieties, the isolation distance for foundation seed is 400 meters and 200 meters for certified seeds. But if the plants are self-compatible and self-incompatible, when we talk about hybrid seed production, that means 25 meters for self-compatible and 50 meters for self-incompatible under certified seeds category. Remember, isolation distance is mainly kept for hybrid under the hybrid seed production condition, where to avoid the unwanted contamination of the pollen, isolation distance is maintained. And this particular criteria of keeping the isolation distance in meters depends on the kind or the nature of that particular crop, like it may be self-pollinated, cross-pollinated or often cross-pollinated. Even the attracting habit of that particular plant's flowers to insects as well as wind also depends on this particular criteria for fixing the isolation distance. So we shall move to the next question. Here we have some other names of particular kind. Okay, so what are the other names? Here directly the things are given, like nucleus seed is called as basic seed, foundation seed is called as mother seed, utility percentage of seed, that is the real value of seed or pure line seed. Impurity of seed is called as dockage. TZ test is also called as biological test. Topographical test or Lancon's test is called as TZ test. Then TZ test is having, or the chemical used for TZ test is 2,3,5 triphenyl tetrazolium chloride. Grodex test. This uses triphenyl tetrazonium bromide. Remember, TZ test involves 2,3,5 triphenyl tetrazolium chloride, but Grodex test includes the chemical triphenyl tetrazolium bromide. Breeder seed is also called as stock seeds. Here you need to remember, nucleus seed is known as basic seed, breeder seed is known as stock seed, and foundation seed is known as mother seed. Next question is, germination occurs when seed moisture is above. The options given here are 40 to 60%, 30 to 35%, 20 to 25% and 20 to 23%. The correct answer is 40 to 60%. The ninth question is, match with appropriate remark. Here the full form or the definition of particular term is given here, given next to it. Dormancy means it is the lack and inactive hydrolytic enzymes. Means for dormancy, lack and inactive hydrolytic enzymes are responsible, means absence of enzymes is also responsible for dormancy. Gibberellins play predominant germination role, that means gibberellins are the predominant germination agents. Cytokinins that lead to initiation of cell proliferation and expansion. Red light, which is at 660 mu, that is for promoting the germination, or it acts as a germination promoter. Then far red light at 735 nm, that is blue red also, it is also called as blue light, that inhibits germination. Next question, that is about formulas. Match with appropriate formulas. The formulas are given directly here. The first is real value of seed. Utility percentage of seed, or pure live seed percentage, that is represented as purity percent into germination percent divided by 100. I repeat, the real value of seed, that is also called as utility percent of seed or pure live seeds, is represented as purity percentage into germination percentage into divided by 100. The second formula is purity percentage, that is represented as weight of pure seed divided by weight of bulk seed. This includes pure seed plus impure seeds, means bulk seed includes pure seed plus impure seed. Here inert matter, weed seeds, other crop seeds are also included, that is multiplied by 100. Third is impurity percentage or dockage, that is represented as weight of impurities into bulk sample divided by weight of bulk sample into 100. We'll move to the next question. Here, matching according to events in different years. This is also already matched. NSC registered in, that is National Seed Corporation. This was registered in 1963 on 7th March. This question is often asked in the exam, so you need to remember along with the date, that is 7th March 1963. Seed Act enacted in 1966, that is 29th December 1966. Third is Seeds Rule notified in 1968. Seed Act enacted in 1966 and notified in 1968. That is Seed Rule in 1968 and Act in 1966. Seeds Rule came into force, okay, in 1969, that is on 2nd October 1969. Essential Commodity Act came in 1955. ISTA, that is International Seed Testing Association, established on 10th July 1924 and it started its function from 1925. Next is again matching. Here different temperatures are given, that is the physical heat treatment, temperature and duration against which disease is used to control. 54 degrees Celsius for 10 minutes is used for to control loose smut of wheat. 54 degrees Celsius for 13 minutes to control loose smut of barley. 38 degrees Celsius for 10 minutes for alternaria blight of wheat control, and 54 degrees Celsius for 60 minutes to control leaf spot of till, that is sesamum leaf spot. I repeat, 54 degrees Celsius for 10 minutes is given for loose smut of wheat. 54 degrees Celsius for 13 minutes is given for loose smut of barley. 54 degrees Celsius for 60 minutes to control leaf spot of sesamum. Then 38 degrees Celsius for 10 minutes to control alternaria blight of wheat. The next question is, in gymnosperms has naked seeds. Dash is the storage tissue. Megametophyte is the storage tissue in gymnosperms, and if it is asked in angiosperms, that is coated seeds. Here endosperm is the storage tissue. Coming to match the following or arranging, 4 things are given here: evaluation, identification, release and notification, and certification. These things are related to varietal release, or whenever a variety has to come to market, this has to follow a proper or scheduled procedure that involves certain steps. So first, what should we do? First we should go for identification, followed by evaluation, then release and notification, and finally certification. We'll move to the next question. Seed certification was started in which country? First time seed certification was started in Sweden. First seed testing laboratory established in Denmark by, the scientist's name is Dr. E. Harlow. Who proposed the production of foundation seed? That was Dr. J. W. Robertson. The 4 options given are Dr. Barley, Dr. J. W. Robertson, Dr. E. Harlow, and Dr. Johnson. And the answer for who proposed the production of foundation seed was J. W. Robertson. The International Crop Improvement Association, renamed as Association of Seed Certification Agency in 1969. The option given here are, it was 1919. Options given here are 1919, 1969, 1963 and 1966. The answer to this particular question is 1919. Next question is, according to Seeds Act 1966, the quality of seed sold for agriculture purpose through 4 options given here are compulsory labeling and voluntary certification. B is voluntary labeling and compulsory certification. C is both labeling and certification are mandatory, and D is none of the above. And the answer is A here, that is compulsory labeling and voluntary certification. Next is, under which section of the Indian Seeds Act 1966 certified seed is certified by a SCA? The section we should name here. The section related to this is section 8. Next question. Contamination by seeds of other crop species is permitted to the small degree up to dash percentage. Options are 0%, 0.01%, 0.001%, 0.1% and 0 to 0.1%. Here the answer is 0 to 0.1%. Next question. Contamination by seed of other varieties of the same crop is generally, options given here are permitted, not permitted, and none of the option. Answer is not permitted because it may create the varietal mixture and confusion among the farmers. Next question is, the total amount of permissible contamination by inert matter and defective seed ranges from dash in case of maize and dash in case of carrot respectively. Here the total amount of permissible contamination by inert matter and defective seed that ranges from, for maize it is 1 to 5%, and for carrot it is 2 to 5%. Coming to next question, match the crops with minimum germination percentage for certification. This is a direct question. Here the germination percentage is already given as matched one. In maize, the germination percentage is 90%, wheat 85%, rice 80%, jawar 80%, cauliflower 65%, bhindi 65%, carrot 60%, and chilies 60%. Next question. Here minimum isolation distance again. For hybrid maize it is already matched. For hybrid maize the minimum isolation distance for foundation seed is 400 meter and certified seed is 200 meter. In hybrid jawar, for other jawar it is 300 meter for foundation and 200 meter for certified seed. For Johansson grass, that is sorghum helipens, the foundation seed isolation distance is 400 meter and for certified seed also it is 400 meter. Coming to bajra, hybrid bajra, it is 1000 meter for foundation and 200 meter for certified. In cucurbits, 800 meter for foundation and 400 meter for certified. In carrot, 1000 meter for foundation and 400 to 800 meter in certified. In sesamum, 100 meter in foundation and 50 meter in certified seed. Next question is, in case of maize, hybrid seed production, border rows, planting practice undertaken too. Means here the emphasis is given on the need of border rows planting in hybrid seed production. The answer is, it provides pollen to a line and it reduces the minimum isolation distance. It is very important. The border rows or planting border rows at the end is the main motto or main reason for providing pollen to a line. A line means that is male sterile or female line. Then other utility is to reduce the minimum isolation distance. Next question is, air screen machine is extensively used for cleaning and grading purpose. We'll move to the next question. Here also it is match the following. Specific gravity separator. This is utilized to divide seeds on the basis of their weight and size. Second is pneumatic separator. Pneumatic separator is used for separating seeds on the basis of their resistance to airflow. Third is spiral separator. This is used to separate seeds based on seed shape. Velvet roll separator. This is to separate seeds based on surface smoothness. Velvet means smooth. Okay. Electronic separator. This is to separate seeds based on the electric properties of the seed. Finally, electronic color separator is to separate seeds based on the seed color. Next question is, here the designated diseases and crops are given and they are matched already. I'll read for your kind information. In case of bajra, that is Pennisetum typhoidis, the designated or identified disease with respect to seed certification is Claviceps microcephala, that is Ascomycetes related fungi, which is a disease. Then Triticum aestivum, here the designated disease is nematode Angina triticide. Then Oryza sativa, paddy bunt. For rice, paddy bunt is a designated disease. Triticum aestivum, for this kernel bunt, that is caused by Neovosa indica, which is also called as cancer of wheat or partial bunt. In sorghum bicolor, that is sorghum, here designated diseases head smut and grain smut. Head smut is caused by Specialotheca liliana and grain smut is caused by Specialotheca sorghai. We move to the next question. The question is, drying generally involves the removal of moisture distributed inside the whole seed. How moisture follows the event? We should talk here. The options given are first capillary action, then evaporation. Next, first evaporation, then capillary action. C is only evaporation and D is none of the above. The answer is first capillary action takes place and then evaporation. That means capillary action is followed by evaporation. Next question is two classes of seed treatment chemicals, disinfectant and protectant. यहाँ पे ऑप्शन दिया गया है एंड फॉर दिस मैं ज़्यादा नहीं पढ़ूंगी. आई विल डायरेक्टली गो टू द आंसर. फॉर डिसइंफेक्टेंट it is a indirect or eradicate the organism present on the seed surface. That means it inactivates the organism present on the seed surface, that is disinfectant. Protectant means that protect seeds from attack by seed-borne and soil-borne pathogens. Next question is matching the chemicals with class of seed treatment chemicals. The answer is given here only. Agrasan GN, that is used as disinfectant. Sirasan is also disinfectant. Thyram is protectant, which is generally used for seed treatment. Captan, that is also used for seed treatment, which is called as a protectant. Then chloranil, that is also a protectant. Agrasan, GN and Serasan, these are organomercurials. Our next question is say true or false. First one is organomercurials are injurious to seeds, particularly to cracked seeds. This statement is true because organomercurials are very poisonous to our body, as well as this may be injurious to seeds. Next is organomercurials are not used in crops like maize, bean, peanut, vegetable seeds, wherever the seeds are consumed directly. For example, if we talk about rice, we remove the husk or we remove the outer part, outer cover. But when we talk about maize or beans, we consume it directly, even many of the vegetables. So if we spray with organomercurials, we may directly consume the seeds. That means the poison will directly enter inside. That is a problem. Then the third statement says organomercurials are used when disease incidence in the seed crop necessitates it. This option is also true, or this statement is a true statement. Next is match specific treatment with specific disease. The different treatments are organomercurials 1%, that is used in dust form to treat the grain smut of bajra. Next is brine solution, that is salt solution, soaking in salt more than 5 to 10 minutes, that is to treat the ergot infection in field of bajra. Next is carboxin solution, that is used to control loose smut of wheat, or carbendazim, that is in dust form, that is also used for controlling the loose smut of wheat. Next question is match with crop and objectionable weeds. Here the objectionable weeds are given directly. In wheat, the objectionable weed is Convolvulus arvensis. In thoria and mustard, Argemone mexicana. Cucurbits, wild cucurbit. Bhindi or okra, wild Abel moisture species. Triticum aestivum, Hirankuri, or that is called as Convolvulus arvensis scientifically. Then thoria and mustard, Satyanashi, that is Argemone mexicana. Shall move to the next question. The question is against which nematode the movement of seed potato grown in the Nilgiri hills to other areas of the country is strictly prohibited. The answer here, the options given are meladogine, that causes root knot nematode. Then second option is Globodera rostachiensis. Third is both A and B, and D is none of the above. The correct option is B, that is Globodera rostachiensis, which is known as golden nematode. Next, some information is given here, that is designated diseases in seed production. In wheat, the designated disease is loose smut, or that is caused by Stylogotriticide. In jawar, there are two designated diseases, namely grain smut and head smut. Grain smut is caused by Specialotheca sorghai, and head smut Specialotheca riliana. In bajra, three designated diseases are important. Ergot is caused by Claviceps microcephala. Grain smut is caused by Polysporium penicilleria. Then green ear is caused by Sclerospora graminicola. Next we'll come to potato. Here many of the designated diseases are present, like brown rot, which is caused by Pseudomonas solanacearum. Then leaf roll, caused by potato virus 1, that is similar to potato virus 14. Mild mosaic, caused by potato virus X, that is similar to solanum virus 1. Severe mosaic, caused by potato virus Y. Scab, caused by Streptomyces cabis. Wart, caused by Syncytium endobioticum. Root knot nematode, caused by meladogine species. Golden nematode, caused by Globodera rostachiensis. Next, in bhindi, the designated disease is yellow mewing mosaic, that is caused by hibiscus virus. In cowpea, three designated diseases are important. Anthracnose, caused by Colletotrichum capsule, then capsicii, then Ascochyta blight, caused by Ascochyta fasciolarum, then bacterial blight caused by Xanthomonas campestis pathover vignicola. In rapeseed and mustard, the designated disease is Alternaria blight, that is caused by Alternaria brassicae. Next question, broken seed, that is larger than half of the original seed and has intact embryo, is classified as. Just concentrate on this question. This is quite confusing type. Broken seed. Okay, seed is broken, that is larger than half of the original seed and has intact embryo. एम्ब्रियो भी है, सीड का कोई स्पेशल फीचर्स डैमेज नहीं हुए हैं, लेकिन सीड लिटिल ब्रोकन है, तो इसको हम क्या बोल सकते हैं? वी कैन कॉल इट एज प्योर सीड. Next question is examination of seed in the laboratory and match with the crop test used. सोरगम के लिए KOH ब्लीच टेस्ट करते हैं, वीट के लिए NaOH टेस्ट करते हैं, सोयाबीन के लिए पेरोक्सीडेज टेस्ट, वीट के लिए फिनॉल टेस्ट, देन वीट बार्ली कॉटन के लिए पेज और पॉलीएक्रिलामाइड जेल इलेक्ट्रोफोरोसिस, जिससे मार्कर रन करके उसके कैरेक्टर्स को चेक करेंगे. नेक्स्ट क्वेश्चन इज द केमिकल 2,3,5 ट्राइफिनाइल टेट्राजोलियम क्लोराइड इज कलरलेस, इंटेंस रेड कलर, ग्रीन कलर, इंडिगो कलर. तो 2,3,5 ट्राइफिनाइल टेट्राजोलियम क्लोराइड एज सच कलरलेस होता है. The next question is the moisture content of seed can be determined by oven method. Thus, weighted seed samples are dried at first option है 130 degree Celsius for 90 minutes, 120 degree Celsius for 60 minutes, 110 or 110 degree Celsius for 90 minutes and none of the above. तो करेक्ट आंसर है द मॉइस्चर कंटेंट ऑफ सीड कैन बी डिटरमाइंड बाय ओवन मेथड. दस weighted seed samples are dried in 130 degree Celsius for 90 minutes. मतलब 1.5 घंटे के लिए बॉईल किया जाता है फॉर 130 डिग्री सेल्सियस. नेक्स्ट क्वेश्चन, थराई डेवलपमेंट कॉर्पोरेशन पंतनगर वॉज एस्टेब्लिश्ड ऑन. करेक्ट आंसर बता देती हूँ और एल्स इट मे कॉज confusion. February 27, 1969 is the right option. Next question is the recommended planting ratio in maize hybrid seed production. The option given are 2 male 6 female rows, 1 male 4 female rows, 3 male 6 female rows and 4 male and 8 female rows. This is for double cross hybrid maize. For double cross hybrid maize, we should follow 2 male and 6 female rows. Next question, generally in hybrid seed production, female parent of seed parent is tagged with yellow label and male parent or pollinator parent is tagged with red label. Next question की तरफ पढ़ते हैं. Here again match crop with seed rate of female and male parents. Okay, मतलब मेल सीड्स कितना चाहिए एंड फीमेल सीड कितना चाहिए देखते हैं. हाइब्रिड्स और डबल क्रॉस मेज में 45,000 सीड्स चाहिए 1 हेक्टेयर के लिए. देन 15,000 सीड्स चाहिए 1 हेक्टेयर के लिए फीमेल सीड्स. 45,000 मेल है फीमेल है सॉरी एंड 15,000 मेल सीड्स है. ओके, हाइब्रिड जवार के लिए दैट इज सोरगम बाई कॉलर 7.5 kg पर हेक्टर फीमेल सीड चाहिए एंड 6 kg पर हेक्टर मेल सीड. इन हाइब्रिड बाजरा दैट इज पेनिसेटम ग्लॉकम 2.5 kg पर हेक्टर फीमेल सीड चाहिए एंड 1.25 kg पर हेक्टर मेल सीड चाहिए. Always remember that the amount of female required is quite high as compared to male because male is only used for pollen supplementation or else female plays very important role in hybrid seed production. Next question को move करते हैं. Say true or false of the following statements. True or false बताना है. मैं statements बताते जाती हूँ. During harvesting of hybrid seeds, male rows are harvested before female rows. मेल रोज को पहले ही हार्वेस्ट किया जाता है फीमेल रोज से क्योंकि कंटेमिनेशन को अवॉइड करने के लिए. ओके, कि अगर मेल रोज पे भुट्टा लगा है if we talk about maize that should not be contaminated with the female one which are fertilized and labeled. इसलिए मेल्स को पहले हार्वेस्ट किया जाता है. Second one is detasseling is done regularly every day as long as tassel emerge on the male parent plants. This is false statement. Moisture content of the maize, jawar and bajra should be reduced to 12%. ये true statement है. Next statement says first field inspections done before flowering to determine isolation, pressure of off-type plants, planting ratios, errors in planting. ये बिल्कुल सही statement है. Next है second field inspection is done during flowering to check pollinating, roguing and male fertile female plants. This is also a true statement. Third field inspection is done prior to harvest and after the seed has matured to reveal its true characteristics. ये भी true statement है. Next is CSCB means Central Seed Certification Board. NSC is initiated in 1961 under ICAR. ये भी true statement है. Moisture content or moisture meter measures the moisture in seeds to an electrical current form. तो ये भी true है. देन एक लास्ट वाला है ट्रू एंड फॉल्स बताने के लिए. So there are 13, still 4 are there. The chief objective of seed production in potato is to produce seeds free from virus Y, X, A and S. तो ये बिल्कुल ट्रू स्टेटमेंट है. Viruses potato Y, X, A and S are transmitted by thrips. ये गलत स्टेटमेंट है यहाँ पे, ध्यान दीजिए. The 11th one is false here. Next statement है potato seed is multiplied at hills during April to August. तो ये true statement है. 2 potato seeds होता है या पोटैटो के डिफरेंट डिसीजेस से बचाने के लिए उसको ऐसे किया जाता है. The main potato crop is taken at plains during November to March. That is true. Next question की तरफ बढ़ते हैं. Which method is specifically used to detect diseases like loose smut fungus in wheat and barley? तो यहाँ पे डिफरेंट टेक्निक्स दिए गए हैं. नेक्स्ट है स्क्लेरोस्पोरा जर्मिनेशन, दैट इज डाउनी मिल्ड्यू ऑफ बाजरा. तो इसके लिए कौन सा मेथड यूज़ करते हैं? ऑप्शंस दिए गए हैं. अगार प्लेट मेथड, ब्लॉटर मेथड, वॉशिंग टेस्ट, सीडलिंग सिम्टम टेस्ट एंड एम्ब्रियो काउंट मेथड. आंसर होगा एम्ब्रियो काउंट मेथड. एक क्वेश्चन क्या बताता है? विच मेथड इज स्पेसिफिकली यूज्ड टू डिटेक्ट डिसीजेस लाइक लूज स्मट फंगस इन व्हीट एंड बार्ली एंड स्क्लेरोस्पोरा जर्मिनेशन, दैट इज डाउनी मिल्ड्यू ऑफ बाजरा. तो इनके लिए सॉल्यूशन क्या है? एम्ब्रियो काउंट मेथड. नेक्स्ट क्वेश्चन में भी ट्रू एंड फॉल्स बताना है. द फर्स्ट statement is kernel bunt caused by Neovossa indica of wheat is prevalent in northern wheat growing states. ये true statement है. सेकेंड है malt extract agar popularly MEA बोला जाता है and potato dextrose agar called as PDA are two nutrient agar media generally used for seed health testing, especially agar plate technique. तो ये आंसर इस सही है. नेक्स्ट द सीड्स आर ट्रीटेड विथ 1 to 2% सोडियम hypochloride, that is NaOCl, and the seed are treated with 0.1% of moisture mercury chloride, that is CHgCl2. ये भी ट्रू है. We shall move to the next statement. Heavy dose of nitrogenous fertilizers increase the disease incidence. ये ट्रू है क्योंकि वेजिटेटिव कैरेक्टर्स और वेजिटेटिव ग्रोथ ज़्यादा हो जाता है. जैसे ही वाटर कंटेंट और लश ग्रीन कलर आने लगता है प्लांट में, तो क्या होगा? डिसीज इंसिडेंस ज़्यादा हो जाएगा. नेक्स्ट क्वेश्चन है 50th क्वेश्चन. ये पूछता है कि सीडलिंग टेस्ट method used to detect. ऑप्शन है these fungi which are capable of producing symptoms on the roots or shoots of young seedlings. मतलब वो फंगाई है जो रूट एंड शूट पे सिम्टम दिखाएगा. द नेक्स्ट ऑप्शन इज those fungi which are able to produce mycelial growth and fruiting structure, then both A and B and none of the above. यहाँ पे आंसर होगा the fungi which are capable of producing symptoms on the roots and shoots of the young seedlings. Next question है what is the male and female planting ratio followed in hybrid seed production of jawar and bajra? तो ये जनरली होता है 2:4. In many countries, new varieties are tested as. Answer said does and does, both A and B and none. तो याद रखिए the answer is N does, that is novel, distinct, uniformity and stability. N does is not operative in India, but varieties are tested in AICRPs. ये बिल्कुल सही स्टेटमेंट है. Clistogamous flowers most efficient floral adaptation for promoting cross pollination. ये गलत है क्योंकि क्लिस्टोगेमी, चास्मोगेमी या क्लोज्ड फ्लावर या क्लोजनेस ऑफ द रिप्रोडक्टिव पार्ट्स, ये सब सेल्फ पॉलिनेशन प्रमोट करने के क्राइटेरिया और कैरेक्टर्स है, तो ये स्टेटमेंट गलत है। नेक्स्ट इज Chasmogamous flowers are most efficient method of adapting for promoting self-pollination. तो ये स्टेटमेंट है false, because chasmogamy क्या करता है, chasmogamy flowers are most efficient adaptation for promoting self pollination. chasmogamy true है क्योंकि chasmogamy self pollination को boost करता है. Next we shall see the types of pollination. ओके यहाँ पे भी मैच द फॉलोइंग, लाइक एक पोल दिया है, वहाँ से मैच करना पड़ेगा किस टाइप का पॉलिनेशन हैबिट है. तो राई है, ये वीट का भाई है लेकिन क्रॉस पॉलिनेटेड है. देन फोलिएज और फोरेज लेग्यूम, ये भी क्रॉस पॉलिनेटेड है. फोरेज लेग्यूम्स होते हैं, कई क्रॉस पॉलिनेटेड होते हैं लेकिन मेजॉरिटी सेल्फ पॉलिनेटेड है. कैरेट प्योरली क्रॉस पॉलिनेटेड होता है. कॉलीफ्लावर क्रॉस पॉलिनेटेड. ऑनियन क्रॉस पॉलिनेटेड. मेज क्रॉस पॉलिनेटेड. बाजरा क्रॉस पॉलिनेटेड. बार्ली सेल्फ पॉलिनेटेड. ओट एंड टोमेटो सेल्फ पॉलिनेटेड. कॉटन एंड पिजन पी और रेड ग्राम आर ऑफन क्रॉस पॉलिनेटेड प्लांट्स. नेक्स्ट है डाइकोगाइमी मीन्स. डाइकोगाइमी क्या होता है, दिस इज मैचुरेशन ऑफ फीमेल एंड मेल एट डिफरेंट टाइम्स, मतलब स्पेशल आइसोलेशन रहा यहाँ पे मेल एंड फीमेल रिप्रोडक्टिव पार्ट्स के बीच में एक ही प्लांट्स के. देन हर्कोगेमी मीन्स. हर्कोगेमी क्या होता है, हर्को मतलब एक बैरियर होगा विच हिंडर्स द सेल्फ पॉलिनेशन. हेट्रोस्टाइली. हेट्रोस्टाइली मतलब डिफरेंट स्टेम स्टाइल लेंथ होता है जिसके वजह से फर्टिलाइजेशन काफ़ी दिक्कत होता है. नेक्स्ट क्वेश्चन इज किसके बगल में क्या होता है, लाइक एग सेल्स आर सराउंडेड बाय 2 synergids. चलाजल एंड हैज 3 एंटीपोडल सेल्स. नेक्स्ट यहाँ भी पूल दिया गया है कि प्लॉइडी नंबर बताना है. जाइगोट का प्लॉइडी नंबर होता है 2n, वैसे ही इंटर्ग्यूमेंट का भी 2n होता है. अगर हम n की बात करें तो एग, स्पर्म, ये दोनों n कंडीशन मतलब हैप्लॉइड कंडीशन में होते हैं, या इसको सिंगल n से डिनोट किया जाता है. देन 3n, ये बहुत ही ज़रूरी इंपॉर्टेंट है, अल्यूरॉन लेयर एंड एंडोस्पर्म दोनों 3n होते हैं. ओके. नेक्स्ट 60th क्वेश्चन है मैच द फॉलोइंग. तो इसमें क्या मैच करना है, इसमें मैच करना है कि कौन सा क्या होता है. ओके. तो टेस्टा होगा आउटर लेयर, देन टेगमन होगा इनर लेयर, हाइलम होगा स्कार ऑन सीड कोट, फ्यूनिकल फॉर्म्स रिच, इसको बोलते हैं राफे, आउटग्राउथ बिलो हाइलम को बोलते हैं स्टाफियोल, कैस्टर एंड नटमेग आर आउटर ग्राउथ्स ऑफ, इसके आउटर ग्राउथ को एरिल बोलते हैं और उसके नटमेग के फ्लावरिंग को एरिल बोलते हैं और कंज्यूमेबल पार्ट होता है वो. देन होल ऑन सीड, ये होता है माइक्रोपाइलर एंड, जहाँ से रेस्पिरेशन या कोई भी वाटर इम्बिबिशन होता है. देन प्रोटेक्टिव शीथ ऑफ प्लूम्यूल, यह है कोलियोप्टेल. देन प्रोटेक्टिव शीथ ऑफ रूट कैप, दैट इज कोलियोराइजा. नॉन लिविंग पार्ट ऑफ एंडोस्पर्म, इसका आंसर है गैलेक्टोमैनर रिजर्व्स. नेक्स्ट क्वेश्चन इज वैरायटी रेजिस्टेंट फॉर फ्यूजेरियम विल्ट इन साइजर एरियाटिनम इज JG11. नेक्स्ट क्वेश्चन, हॉलो ब्लाइट डिफेक्ट इन पीस एंड खायरा डिसीज इन राइस ड्यू टू डेफिशिएंसी ऑफ डैश एंड डैश रेस्पेक्टिवली. तो पी के हॉलो हार्ट के लिए बोरॉन ये होता है रिस्पॉन्सिबल होता है, एंड खायरा डिसीज in rice is due to zinc deficiency. So finally we shall see some of the seed multiplication ratios. हालांकि मैंने सीड मल्टीप्लिकेशन रेशो पे एक डिफरेंट वीडियो बना दिया है. यू कैन डायरेक्टली विजिट टू डॉक्टर वर्षा गायतोंडे's चैनल. और इम्पॉर्टेंट क्रॉप्स के लिए यहाँ पे मैंने सीड मल्टीप्लिकेशन रेशियो जस्ट मेंशन किया है. फॉर बाजरा द मल्टीप्लिकेशन रेशियो इज़ 1:200. मतलब एक सीड अगर आप डालेंगे तो उससे आप कितने सीड्स हार्वेस्ट कर सकते हैं, या कन्वर्टेबल प्रोडक्ट होता है सीड मल्टीप्लिकेशन रेशियो. तो 1:100 मल्टीप्लिकेशन रेशियो होगा रेपसीड, रेड ग्राम, हाइब्रिड मेज, सोरगम एंड जूट. पैडी में 1:80 होगा, वीट में 1:20 होगा, चिकपी में 1:10 होगा, पोटैटो में 1:4 एंड ग्राउंडनट 1:8. Friends, I think the information furnished by me is helpful to you, and if you subscribe my channel or if you keep on commenting or liking, it will be a big motivation for me to make further useful and informative videos for you. Thanks for patient listening and watching. Thank you once again.